Hi everyone, it's me. We're back again for another lecture. Last time we were talking about different types of muscles, focusing on skeletal muscle, and we're going to continue today talking about skeletal muscles and how they contract. And so if you remember last time we talked about actin and myosin, and this is a diagram here. It's hard to see, but this double kind of helical structure is the actin. This is myosin. Part of myosin has these things called little head groups that stick out from the myosin, and they're the thing that's going to be the part that's the motor protein. They're going to grab onto actin and pull. Okay, and so I just wanted to show this because it's kind of neat to show as a kind of a little video. This is actually what's happening. So again, myosin, this part that's the motor protein, is going to break down ATP, and using the energy from ATP, it's going to grab onto actin and pull. It's going to walk along actin, and that's what a motor protein does. It uses the energy from ATP to generate movement. And we're going to get back to this in a little while and look at the actual other parts of this picture. But for now, I want to actually go back to where we finished off. And we were looking at this kind of cartoon type of thing of the sarcomere. And the sarcomere, remember, is the contractile unit of a muscle, and this skeletal muscle here is arranged in different types of proteins that are interspaced between them. We have myosin, which is this thick filament, and that's the motor protein. And then we have actin, which is this thin filament. It's actually a microfilament. And that's kind of just like a rod that's going to be moved by myosin. And when a muscle is going to start to contract, we have the sarcomeres, which are demarcated or something, whatever that word is, have boundaries called the Z lines. And the Z lines are going to actually get closer together, and the whole sarcomere is going to shorten upon contraction. And note that the myosin does not change its length and the actin does not change its length. All they do is increase their overlap upon contraction. Okay, so we're going to actually look at this in more detail on the molecular level and what's really happening with that myosin and that actin. And so here is another cartoon showing the same picture as we showed before, myosin in this pink color and actin in the light blue. But now it's a little bit more detailed. And what you can see here, if you look carefully, are these little blobs that are attached to the myosin. And those are called myosin heads or myosin head groups. Each one of those myosin heads is an enzyme that can break down ATP. So it's an ATPase. So there's ATPases that are part of myosin along the whole length of myosin. And that's what makes this thing a motor protein. It's going to use the ATP and it's going to generate movement of the actin by walking along the actin. But it's not just doing it at one place, it's doing it throughout the whole myosin molecule along all of these myosin head groups. So what we're going to do is I'm just going to look at one of the myosin head groups and see how it is going to move actin. So here we have actually one of those myosins that are now zoomed in. Sorry about that. Zoomed in. And we're going to look at it in a little bit more detail. So as we said, here's that myosin head, and it's looking it above it is the actin. The goal is going to be for the myosin to grab onto actin and pull it this way, okay, in that direction. And then at the same time, you have all these other myosin head groups on the other side that are doing the same thing, and they're pulling in the opposite direction so they're going towards each other. But let's just look at this one for now. So the first step in this kind of cycle is going to be the myosin has to bind ATP. That is absolutely required for the initiation of contraction, but it's also required for myosin to let go of actin from the previous contraction. That sounds weird. Yeah, actually, myosin will be stuck, attached to actin, unless it binds another molecule of ATP. And so that's what's going to happen. Another molecule of ATP is going to bind, and myosin will just bind the ATP, let go of actin from the previous contraction, and then it will sit there in what we call a low energy configuration. And it'll sit there until it hydrolyzes or breaks down the ATP, which will provide it the energy to actually cock its head. So upon hydrolyzing this ATP, the myosin then uses some of the energy to cock its head like this. Okay? It hasn't grabbed onto actin yet, but it's in this high energy state. And it's going to be waiting to see if it can find a place on actin to bind. And when it does, that's the next step. The next step is going to be to cross-link. So if it finds a spot on actin that is available, it's going to cross-link and form a cross bridge. And then as soon as it does that, it's going to do what's called the power stroke. It's going to pull, and it's going to move actin this way. And that's the final step, okay? And the thin filament or the actin moves towards the center of the sarcomere, which moves this Z line closer to the other Z line, which is over here. And the other Z line is moving this way because remember, there's going to be these myosin head groups on both sides, and they're pulling towards each other when they bind the actin. And that's how a muscle contracts. Okay. Now, we're not done yet, though, because the last step after that power stroke is you've got to bind another ATP. And what will that do? That will allow myosin to let go of actin again. So again, in order for myosin to let go of actin from the previous contraction, it has to bind another ATP. Now, why is that important? Well, it turns out it doesn't seem to be that important for us when we're alive because we always have a continuous supply of ATP as long as we are aerobically respirating. And so following any kind of contraction, boom, we bind another ATP and myosin's ready to go in a low energy configuration, ready to go, okay? But it does become a problem upon death because when an organism dies, it depletes its ATP stores pretty rapidly. And so what will end up happening is any contraction that has happened can't let go. Okay. And so because it can't let go, you end up getting what's called a rigidity of the muscles, which is called rigor mortis. So I'm going to write that down. Rigor mortis. Rigor mortis literally means rigidity following death. And that occurs because there's no more ATP and then myosin cannot let go. It's stuck in this position right there. And it causes an intense rigidity which doesn't happen right away, but within about 4 hours, depending on the temperature, it begins. And within about another 8 hours or so, you're in full rigor, which means that all of your muscles, with not just the muscles that you can see, but the muscles internally, it's all very rigid. And it's so rigid that it's very, very hard to break that rigor. So if you're stuck like this, a pathologist will not be able to open up your hand. It's almost like they have to break it open. Okay. Eventually, the rigor subsides within about 48 hours total because the body starts to decompose and bacteria start to decompose and break down these proteins. And that actually just breaks that linkage there as these proteins begin to break down. Okay, so one thing I've always thought about was if you have somebody that has gone into rigor and their finger was like this is kind of morbid, how could you get it to move up? Well, besides trying to break it, you could actually inject ATP. And if you inject ATP, you might be able to, if you inject enough of it, get it to open up. Although it might come right back down, but you could get it up if you wanted to. Okay. And then you could just, if you wanted to, clamp it there until rigor subsided. So I just wanted to bring that as a kind of a thought question, that it is ATP that's causing, the lack of ATP that's causing that rigor mortis. Okay. And we're going to get back to this and why it actually does end up contracting even when you are upon death, because when you think about it, if you have ATP all the time, which we do as long as we are alive, then we would always have this happen, then we would always have this happen, we'd always have this happen, and we would always have this happen. And so, just based on what I've shown you, as long as you have ATP, you should be constantly contracting and relaxing, contracting and relaxing. We don't do that though. So there must be something else. There must be something else that controls this whole contraction, because we always have ATP. So as long as myosin is able to grab onto actin, you should always have contraction and then relaxation and then contraction. But we don't. And so, as we're going to see in a little while, the reason why we don't constantly contract and contract and contract with constant amounts of ATP and let go and contract and let go is because the actin in our cells is not accessible to the myosin. So remember here, if you look back here, in order to form that cross bridge, you've got the ATP, okay? Everybody has ATP, so they can let go from the previous contraction. The myosin can do the cocking its head, but then the actual next step was to make the cross link or the cross bridge. And you can only do that if this actin is accessible to the myosin. And it turns out that it's not accessible. There's going to be something that's covering the actin and keeping it masked until you really want to contract. And then when you really want to contract, you will uncover the actin and boom, you get that. Okay? So just hold that thought and we'll come back to that later on when we actually look at this mechanism more detail, because there is something else needed to regulate this contraction besides ATP. Okay, so that's that for now. What I'm going to do now is kind of shift gears. We'll come back to this in a little while. I want to shift gears because it turns out that in order for a skeletal muscle to contract, it must be stimulated by a nerve. Okay, so skeletal muscles will only contract if they are stimulated by a specific type of nerve. And that nerve is called a motor neuron. Neuron is the name for nerve cell, okay? And so the specific type of neuron or nerve cell that will stimulate a muscle is called a motor neuron, okay? As we'll see later on, the other types of muscles that we introduced before, cardiac muscle and smooth muscle, don't necessarily need stimulation by a nerve to contract, particularly cardiac muscle. It actually contracts automatically within itself and doesn't need nerve stimulation, but skeletal muscles do. And so they have to be stimulated by something through the nervous system. So I'm going to introduce a little bit of the nervous system now. And how it is organized. We're not going to go into detail here. We're just going to talk about the part that is necessary for a muscle cell to contract. And so the way the nervous system is organized, it's organized into two main groups of nerves. Nerves that are part of the central nervous system, right here, and nerves that are part of the peripheral nervous system. Now, the central nervous system is defined as the brain and the spinal cord. That's what the central nervous system is, the brain and the spinal cord. The peripheral nervous system is the nerves that extend out of the brain and spinal cord. Those that are on the periphery, that is the ones that go into the brain or spinal cord, and ones that come out of the brain or spinal cord. Whereas the ones within the brain and spinal cord are part of the central nervous system, and they're the ones that are integrating and taking information from the outside world that's coming in through sensory input, integrating that information, deciding on what to do, and then telling the rest of the nervous system maybe to put out some motor output. So, for example, this loop right here involves something called a sensory receptor that would be on the surface of your skin. You've got these sensory receptors, like touch receptors or pain receptors or whatever. And here is something that's going to cause you a stimulus, some kind of sensory input, some pain, okay? You get bitten by a mosquito and you immediately get some sensory input into the brain and spinal cord. They decide with neurons inside there to send an output through a motor output, which says slap the mosquito and then start scratching, okay? So that is a loop right there where we involve the peripheral nervous system that is coming out of a receptor from the outside world that's sensing information, integrating in the central nervous system, and then sending an output that in this case is some motor output. Okay, it doesn't have to be motor output. It could be causing glands to secrete things or something like that. But in this case, when we're talking about a skeletal muscle, it's going to be a motor output where your hand is going to hit your forearm, your upper arm, okay, and to try to kill that bug. Okay, so let's look at this a little bit more detail with the nerves that are here, the peripheral nerves and the nerves of the central nervous system inside here. And so we've organized it into 3 different types of nerves. And that's shown here. So here is the outside world here where you're getting sensory information through a receptor, and a sensory neuron, that is a nerve that receives information, sense information, is going to be the first step. It's going to receive that information. It's going to send that information to the central nervous system, which is going to contain potentially another neuron. This time it's called an interneuron. So interneurons are neurons that are in between the sensory system and the motor system. And then finally, the interneuron, if there is an interneuron, is going to send the information out through and stimulate a motor neuron, which is going to cause your movement. Okay, so that's that loop right there that we saw here, but with the different types of neurons. Okay, now I'm going to show you an example that you will not need to know. And this is an interesting example because it shows this whole loop right here in one example where you have all 3, and in another example where you actually don't have an interneuron, you just have the sensory, and it actually comes in and connects directly to another neuron. So let's look at this. And what I'm talking about here is the knee-jerk reflex. Okay, the knee-jerk reflex is shown right here. When you go to a doctor and a doctor takes one of those rubber hammers and he or she hits your knee, what they're doing is they're trying to stretch a particular tendon that is in your kneecap. It's called the patellar tendon. And that stretch is sensed by stretch receptors or sensory receptors, and they send information to a sensory neuron. Okay, so that's the first step and the second step. Now, what happens then is 2 things happen. What you want to do is you're going to want to, the end result is going to be to move this lower leg up. And so this patellar bone is actually attached through tendons to this bone right here. And so you're going to want to contract this muscle, okay, and you're going to Um, Once you contract this muscle, which is the quadriceps muscle, that muscle is going to want to contract. That's going to move this whole thing up. But at the same time, you want its antagonistic muscle to relax. You can't have them both contracting at the same time, otherwise you wouldn't do anything. Okay, so you want one muscle to contract, that is this muscle, and you want this muscle, which is your hamstring muscle, to relax. And so this sensory neuron actually splits and does two things, which is kind of cool. The first thing it does, well, it's not in any necessary order, but one thing it does is it stimulates directly the motor neuron that causes the quadriceps to contract. So in that case, there is no interneuron. It just goes sensory neuron right to stimulating the motor neuron, and that quad contracts. But you also want to prevent this muscle from contracting. So that same sensory neuron splits off and stimulates an interneuron in the central nervous system. They're both in the central nervous system, but here we have an extra neuron, one of those interneurons. And this interneuron actually doesn't stimulate this motor neuron, it inhibits it. So that's kind of cool. So you've got this sensory neuron doing two things, indirectly inhibiting this motor neuron and directly stimulating this motor neuron. And you end up contracting this and making sure this one doesn't contract and it stays relaxed. And that moves the leg up. So that's how this knee-jerk reflex works. Now, what's also interesting is this reflex here does not involve the brain. It involves the central nervous system, but not the brain, the spinal cord part of the central nervous system. Okay? And it has nothing to do with the brain. In fact, you could actually be brain dead, okay, and you could still potentially have the patellar reflex. Okay, so people that are officially brain dead, or they're in a coma and they have no brain activity at that time, okay, they're not measuring very much brain activity and they're in a deep, deep coma, they can still have this reflex through their spinal cord. And even somebody that is really dead but is being kept alive by artificial means can still have that knee-jerk reflex because it's only involving the spinal cord and not the brain. But again, that is something you don't need to know, but it just is an example of showing that. I just thought it might get you interested in this. So now we're going to look at this motor neuron in detail and what is actually happening to make it contract a muscle. So now let's look at a picture of a motor neuron. Now this comes from your book, and I like to actually draw things out, which you know because you've seen me draw it on the whiteboard, but I'm going to draw it out in the next picture just so that you can draw it out in your notes and label everything here, because this is just a little bit kind of too complex. But basically, this is the structure. There is a receiving end of a motor neuron, a part of the motor neuron which is just like a typical cell called the cell body, and then there is a transmitting end, and then there's terminal to the ending. We're going to go through each parts of this right here, but the labels are right here, and I'm just going to show it now on my little picture. So this is my drawing, which you could actually draw this one if you want in your notes, or not draw anything at all, or you could draw the way that I drew it because I like mine better, only because, you know, I'm biased. Anyway, so this is an anatomy of a motor neuron, and we're going to go through each one of these things right here. But now let me just show you and just describe things in order. So we're going to start off with this, these little extensions that come out of the typical cell part of the motor neuron. And these are called dendrites, okay? And dendrites are going to be the receiving end. They're going to be the ones that are going to get the information from, say, a sensory neuron or from an interneuron, okay? They're going to receive information. That could be inhibitory, it could be stimulatory. Then you have the cell body, which, as you can see here, is a typical cell. It's got nucleus and DNA. It's got smooth ER, rough ER with the ribosomes. It's got mitochondria. It's got a lysosome here, Golgi. And it's got these things called vesicles. So if you look right here, these are vesicles. They're secretory vesicles that are going to actually move down this extension of the cell body called the axon. So the axon is going to be the transmitting end of a motor neuron, and it's going to do two things. One thing is it's going to allow for these little vesicles that are carrying chemicals to move down along the axon. And they actually move along microtubules. Remember that vesicle transport. So microtubules are carrying those vesicles down through this axon. And that's going on all the time. And those vesicles are then going to lodge themselves in this terminus, this end, which is an extension of the axon. And this terminus down here is actually called the synaptic terminal. Okay. And now we actually call these vesicles, instead of just calling them secretory vesicles, they're called synaptic vesicles. And the reason for that is because there's going to be a region right here, okay? And that region is going to be a paper-thin space between this motor neuron and the muscle cell. And that paper-thin space is called the synapse or the synaptic cleft, same thing. And that's why this is called the synaptic terminal and synaptic vesicles, because they're at the point right next to the space, which is called the synapse, between this nerve cell and the muscle cell, which we haven't shown yet. We also have on this something called the axon hillock, which is the beginning of the axon, and I'll describe what that is in a minute. And then we have something called a myelin sheath and these nodes of Ranvier, named after Ranvier. Okay, so let's go, and I actually typed out the different parts of the anatomy and described very briefly what they are. So let's go through them and we'll come back. So first of all, the dendrites. That's the receiving end of the neuron, those little fibers that extend out of the cell body, which is like a typical cell. So there's the dendrites, there's the cell body. Okay. Then we have the axon, which is the long transmitting end of the neuron. The axon can be very, very long. In fact, in your sciatic nerve, which goes from the base of your spine all the way down to your feet, that axon can be almost 3 feet long, depending on your size. And me, it's like a foot and a half. But if you're tall, it can be very long. So that's the transmitting end of the neuron. And then we have this thing called the axon hillock, which I said was the beginning of the axon. And that is actually the place where a decision is made to either send a signal down the axon and transmit a signal or not. And as we'll see in a moment, that signal is going to be all or none. You're either going to send a signal or you're not. And if you do send a signal, it's going to be the same strength of signal as any other signal. They're all the same strength. But the axon hillock is where the decision is made. And that decision comes from all the information that's coming into the cell via the dendrites. So it could be, you could have stimulatory information, you could have inhibitory information, and that's all integrated at the axon hillock. And if enough stimulatory information is gotten there, then a signal is sent down. Okay, and so moving on to the next part, we have the synaptic terminal, which is the end of the axon, and the synaptic vesicles, which are carrying a chemical called a neurotransmitter. So here's the synaptic terminal, and these vesicles are carrying something in there. You can see it's little red dots, and those dots are a type of chemical called a neurotransmitter. And we're going to talk about a couple of neurotransmitters today. Okay, and so that's what that's doing. And then we have myelin and these nodes of Ranvier. So I want to talk a little bit about that myelin in a little bit, but basically what the myelin is, it's a lipid insulator that's going to surround the axon. And the nodes of Ranvier are in between the lipid. They're like concentric circles that go around, like sort of like a Swiss roll over the axon. It's like an insulation of a wire, but there are spaces between the lipid insulator cells. And those spaces are called the nodes of Ranvier. So at the nodes of Ranvier, there is open axon and no lipid insulator. Okay. And we'll get back to myelin and the nodes of Ranvier in a moment. But first, let's go back to what is happening at the axon hillock. I kind of alluded to it and talked a little bit about it, but what's happening, as we said, was information is coming in from all of the dendrites. It's collected at this axon hillock. And if enough stimulatory information comes in, then the axon hillock meets a threshold. That threshold of stimulation is at some point, and as soon as it hits that, even if it goes over, it doesn't matter. As soon as it hits that threshold of stimulatory information, a signal is going to be sent through. And that signal is called an action potential. And what an action potential is basically electricity. So how does that work? Well, we go back to this axon and the axon hillock. In the axon hillock region, there are a lot of channels, ion channels, and those ion channels are specific for sodium. So there's sodium ion channels there. And it turns out that, like any other cell, sodium is present much more on the outside of a cell than on the inside. And that's in every cell, whether it's a neuron, whether it's a muscle cell, whether it's one of your skin cells, it doesn't matter. The outside of a cell is much higher concentration of sodium than the inside. So if you were to open up channels specific for sodium, sodium's going to rush in. Okay? And as that sodium rushes in here, okay, it's going to diffuse towards this region right here. It's going to go down. And that's where we're going to start the signal. So you're going to get a massive influx of sodium here via those sodium channels. And as those sodium ions rush into the axon, they move rapidly down the axon, and that is electricity. Okay. Now, as the sodium rushes down, okay, we go back to here, rushes down, it turns out that there are voltage-gated sodium channels. Remember channels, and remember gated channels, and remember voltage-gated channels are ones that are opened or closed with electricity. And what is electricity? The movement of ions. And so what happens are there are voltage-gated sodium channels in these regions of the nodes, okay, where it's opened up, where there's no insulation. So there's voltage-gated sodium channels there. There are voltage-gated sodium channels there, and there, and there. And what happens is as that sodium moves down very quickly from here, that's voltage. And they open up the channels here, and more sodium rushes in, and more sodium goes down. And it opens up the channels here, and more sodium rushes in. And more sodium rushes in through these voltage-gated channels, which are opened up by the sodium inside. And so it's almost like a positive feedback. As sodium goes in, it opens up its own channels, and more sodium comes in and goes down and opens up more and opens up more. And it's very, very fast. It could be greater than 200 miles an hour, that signal of electricity. And the reason why it's so fast is that you only have the channels at these openings. And that's where massive amounts of sodium rushes in. And as the sodium comes down here, instead of having channels everywhere, you actually put insulation around the rest of it. So you get no any kind of leaking out. So the insulation in the myelin insulation is there to prevent any kind of movement in and out of any kind of ions. And then the only place where you can get in or out is at these nodes where they're going to go in because the channels cause them to go in through facilitated diffusion. And so literally it goes massive amounts here as it moves down, more in here, more in here. So it's almost like these jump. The signal jumps and goes very, very fast. Okay. So for what you need to know is that the nodes are the exposed part of the axon where they're having channels where sodium rushes in and kind of perpetuates that signal very, very quickly. Okay. Now, let me go here. Okay. Now, what I want to talk about now is a little bit about that myelin sheath because we finished here. Let me finish this actually right here. So the channels are the only places where you have the nodes and you get that very rapid movement because it kind of almost jumps as it goes more and more and more and more. What's going to end up happening is as you move that electrical signal down this axon, that electricity is going to spread into the synaptic terminal, into all of these little termini. And that's going to eventually cause these vesicles to exocytose. And they're going to release their contents into this little space. And the contents are neurotransmitter. So the vesicles don't actually move into the space. They actually fuse with this membrane right here and exocytose their contents into this space. So that's the end of the motor neuron's responsibility. Okay. And so let me go back to this myelin sheath before we start going on to what happens next with the muscle cell. And so that myelin sheath, as I mentioned, was an insulation. It's a lipid. Myelin is a lipid that wraps around the axon along the entire axon except for those places where the nodes are. And most axons are myelinated. And what the myelination does is it insulates the axon and it actually allows the propagation of that electrical signal to go much faster than if there was no insulation and you might end up with some leakiness or something. And it turns out that sometimes you do have situations where the myelin starts to go away. And those conditions are called demyelinating diseases. And what they do is they cause a destruction of that lipid insulation, the myelin, and what it ends up doing is leads to inefficient transmission of that electrical signal. Sometimes the electrical signal just stops, okay, because you can't get that rapid movement of those ions because there's no insulation there. And so I just want to quickly talk about one example of a demyelinating disease that you may have heard of, and it's called multiple sclerosis, or MS, which I want you to know. And the reason why I want you to know this, besides the fact that it's relevant to this lecture, is that multiple sclerosis is something called an autoimmune disease. And we're going to see a number of autoimmune diseases throughout the rest of the course. An autoimmune disease is a disease or a disorder in which your own immune system, that's what auto means, self, your own immune system attacks itself, attacks something within your own body. Sometimes your own immune system will produce antibodies that will directly attack something. Sometimes it will produce something else that will degrade something. So it's your immune system. It could be white blood cells. It could be anything having to do with the immune system, like antibodies attacking a particular part of your body. It attacks cells. In this case, you're going to generate an immune reaction against your own myelin, and it's going to degrade your myelin. And so individuals that have MS, they will generally, in fact, it turns out that MS does not affect the motor neurons, even though it does affect movement. it actually affects the neurons in the brain. And because of that, you can't get the signal to the motor neuron to cause movement. But I still wanted to bring up what myelin and multiple sclerosis was. It's an autoimmune disease where your body attacks the myelin that surrounds certain axons. And it leads to a devastating effect. For some, it could lead to early death, but some people manage it quite well. I know several people that have MS, and it does tend to run in families like all autoimmune diseases. It's not a specific genetic gene or something, but once you have an autoimmune disease, you often have other autoimmune diseases in your family. And so we're going to look at a couple of other ones later. But generally, what happens is the individual has problems with motor movements due to the breakdown of the myelin in the brain neurons. And eventually, they could end up in a wheelchair. They can also have respiratory problems from other nerves, other systems that are not working well due to the degradation of the myelin. But we'll talk about some more autoimmune diseases later. MS, as is the case of most autoimmune diseases, is exacerbated or gotten worse by stress. So individuals that have MS are try to keep a lower stress levels in their body, and they could go into what's called remission for many, many years where they have no symptoms at all. And then some kind of devastating thing happens to them, like a tragedy, and they start seeing the symptoms again. So it can be induced by stress in individuals that have the MS to start with. Okay, so I'm going to stop there and we're going to finish the lecture next time. And what we're going to do next is actually look at how the motor neuron here is going to connect through a very, very small, small space with a muscle cell and cause muscle contraction. Okay, so we're going to stop there and I'll see you later. Bye.